Hi, first of all, I would like to thank the organizers for giving me the opportunity to give this short talk. And I want to talk a bit about our recent efforts on expanding our knowledge about some low represented weird plant genomes, where I work here at the Max Planck Institute for Plant Breeding Research. And as you see, we actually work in plant species, tropical plant species mainly from South America and Brazil, which have the so-called holocentric chromosomes. And I want to talk a bit about these very interesting genomes today. And basically, I mean, there are like two main different types of chromosome organization based on the centromere distribution of those structures. And basically, we have monocentric chromosomes, which have like a single centromere per chromatid. And we also have many species actually that have holocentric chromosomes. And basically what we see that in mitosis, microtubules attach along the entire length of the chromosomes, and they have this so-called holokinetic activity, which then makes those chromosomes in anaphase to migrate like in a parallel configuration instead of this V-shaped configuration that we typically observe in monocentric species. Okay, so actually monocentric species are quite common in several clades, and they independently originated in plants and animals multiple times. In plants, they are actually present in both dicots and monocots. And today I'm mainly focused on this group of species, so the Juncus and Cyperaceae family. But we're also now expanding our efforts to sequence like multiple genomes of other holocentric species and other interesting groups. But they are also found in many animals. For instance, in many insect clades, we find them quite common. Okay. So why to study holocentric chromosomes? Basically, in my group, we are focused on this group of species because centromere distribution can actually impact on genome architecture and regulation, genome evolution, and also in cell division and also meiosis, which are also interested in understanding these adaptations in my research group. Okay. So for this study, we try to first characterize like several holocentric genomes of three Brazilian species of the Rhynchospora genus. So in Rhynchospora, we find the so-called repeat-based holocentromeres, which means that each centromeric unit in this species has a specific site of a repeat sequence. And what was very interesting, because before in the literature was proposed that Juncus and Cyperaceae, they have a common origin to transition to holocentricity. But recently it was proposed that actually Juncus from the Juncaceae family is a monocentric, not holocentric species, which brings this as the closest monocentric relative to our Rhynchospora species. So we decided to sequence like all these four species, and here is very interesting to see that pubera, which shares the same chromosome number as Rhynchospora breviuscula, has actually a 4 times larger genome. And Rhynchospora tenuis has only 2 chromosomes, which is the lowest chromosome number found in plants, which also shares a similar genome size to breviuscula. And Juncus effusus, which is a monocentric with a small genome size and a high chromosome number. So basically, after sequencing these first genomes, we found like very interesting differences between in the genome organization of these species. So basically, we found that transition to holocentricity has greatly influenced the genome architecture and also the distribution of the main genomic features. So in Juncus, for instance, which has monocentric chromosomes, the high C pattern is very typical for a monocentric species with these high interactions between long-distance high interactions between the 2 chromosome arms, and also some kind of interactions between different chromosomes. When we see the sequence distribution in these chromosomes, basically we see that genes are mainly clustered to chromosome arms, like typical for monocentric species, and those repeats in heterochromatin tends to be more constricted at regions close to the centromere. But very interesting, when we sequence the genomes of Rhynchospora, and here I'm showing for pubera, but that's the same thing that we see for all the 3 species. So basically what we see that first at the high C interaction matrix, we don't see anymore those long-range interactions between distant regions in the chromosome. So basically, we show here that there is no interaction between two chromosome arms, and that's because holocentric chromosomes, they actually have no chromosome arms. They have a single entity where chromatin contact seems to be more or less uniformly distributed along the whole extent of the chromosomes. And also very little interaction between different chromosomes was observed. Very interesting, also the sequence distribution, all kinds of sequence distributions also shows this uniform pattern. So all genes, centromeric repeats, like the case of the TIBA repeats, and heterochromatin and euchromatin is also uniformly distributed. So you can check more detail of this if you want to see in our recent publication. And basically here we're showing that this transition to holocentricity has a great impact on the genome organization. So what is also very interesting in holocentric species, because they can also tolerate chromosome breaks and fusions in contrast to monocentrics, because centromere activity is actually dispersed along the whole genome. And that was very interesting because we could actually verify this after sequence those genomes. And what we could show here, basically that this along the evolution of those species, we found like a very interesting pattern of chromosome fusions in the Rhynchospora genus. So basically the ancestral karyotype is proposed to be n equal 5. And we found now that the Rhynchospora tenuis that has this very low chromosome number, it's actually resulted from 3 chromosome fusions, end-to-end chromosome fusions that reduce it to 2 chromosomes only. Rhynchospora breviuscula likely maintained a similar karyotype as the ancestral one. And very interesting and unique here is the case of Rhynchospora pubera. So Rhynchospora pubera shares the same chromosome number as breviuscula, but in fact has 2 rounds of whole genome duplication followed by several cycles of chromosome fusions, which actually reduces this chromosome number back to the ancestral chromosome number m equal 5, and were actually masking, hiding these 2 rounds of whole genome duplication that we could not suspect until we sequenced at chromosome level this genome. That was very interesting. So now we're actually interested in extending this analysis to other species in this group, because we found now that holocentric chromosome seems to have a rapid evolution by both chromosome breaks and fusions. So now we are trying to expand and sequence more like Brazilian Rhynchospora from different clades, so clades that have a very high chromosome number, and clades that have high chromosome number variation, and other clades that are more stable. And what actually find here, the first genomes that we're now expanding our analysis, basically that we see that both fusions happen here in the pubera and tenuis as previously shown, but now we also find that fissions, chromosome breaks, can also occur at a high frequency. And basically we see that the high chromosome number found in 2 other Rhynchospora species basically resulted from chromosome breaks. And we also sequence like a basal genome in the family that we're also trying to investigate if it's also monocentric or holocentric. And basically what we see here that there is a very high conservation of syntenic blocks. Also, what's interesting that there is a very negative correlation between chromosome number and genome size. So the higher the chromosome number, the smaller is the genome size that we find so far. So just showing you like a brief summary of our research and how these genomes are very diverse, and they can really actually bring us like new ways how chromosomes and genomes can evolve. And that's how we are now trying to expand a bit in this analysis. So with that, I'd like to thank you, and thank you my group and also my department and all the fund that I received to do this research. And that's it. Thank you very much.